parameters. For example, Deinococcus, there is a very known bacteria that is radio resistant, but we have other stremophiles, for example, this hyperthermophile that can survive up to 122 degrees. So it's very adapted to these conditions. And some other examples here of these recordists of stremophiles. And when we think about these stremophiles, it's very interesting because they have a lot of survival strategies and adaptations to these extreme conditions. Here in this video, we have only an example of a psychrophile that is adapted to low temperature, and they have these ice nucleation proteins that are capable to change this physical state of water from from liquid to solid instantaneously, as we can see here. This is a product of a protein that are produced by these psychrophiles. But several strategies, survival strategies of these stremophiles are still unknown. And why is it important to study these stremophiles here on Earth? Because studying these stremophiles, we can define the limits of life in a planetary context. So here in these figures, we have some parameters, actually 4 parameters like pressure, pH, temperature, salinity. And the first here, we can have a big concept of what are the limits of life related to this parameter. And we have basically life surviving in almost all values of pH, salinity, but it seems that temperature is a narrow range that life can survive, and high pressures is another parameter that life might be sensitive, at least life as we know. And this is very important because we can define, according to these limits that we know, we can define habitability of other bodies of our solar system or even beyond. So it's very important to define these limits of life as we know it here in our planet. So we are studying here in our group specifically extreme marine environments. So you might have known that we have different extreme environments on Earth, like deserts, caves, polar regions. But what we are talking here now is about the marine extreme environments. Some examples we have here is the hydrothermal vents, some polar volcanoes that we can observe that have marine influence, and the deep sea. So it's specifically the environments that I will show to you today. And why this is important to study these marine stremophiles? You might have heard that it's a very hot topic in astrobiology is the presence of these ocean worlds in our solar system, and where we can observe evidences in some icy moons of our solar system that in these moons they have like a liquid ocean below a thick layer of ice. And this ocean, a lot of evidences have shown that they have keystones of habitability, not only water, liquid water, but also energy and elements that could provide not only evolution of life, but also could provide characteristics for the origin of life itself. And we have some examples here like Europa, Enceladus. There are very important targets for the future missions to search for possible life forms, stromophilic life forms in these oceans. And how can we compare these analog environments when we think about these marine ecosystems? So here we have a general example of the deep sea on Earth. So the deep sea on our planet is the biggest and the most unknown ecosystem on Earth. And the first question we have to do is, how do these microorganisms survive under these extreme pressure conditions or extreme low temperature conditions? And another question is to understand, how do they obtain energy in this ecosystem? So how, which metabolisms they are performing to obtain energy in these marine ecosystems? And based on these questions, we can try to identify potential life forms in these extraterrestrial analog environments, as in these icy moons, these ocean worlds, and search for biosignatures and propose novel biosignatures based on what we know here on Earth that are thriving in these marine extreme ecosystems. And here I have some examples that we are studying in our group. The first one I show to you is a cave. Obviously, it's not a marine ecosystem, but just an example that we start to use molecular tools to review and propose novel functional models of these extremophiles using these molecular techniques. So I'll show to you how can we look for these novel marine extremophiles in these ecosystems and trying to propose novel biosignatures for these marine ecosystems on Earth. When we think about culturing these microorganisms, basically around 1% of these microorganisms can be cultured in laboratory conditions. Almost all of these marine extremophiles are still uncultured. For this reason, we use basically molecular methods. So we can basically we take these samples and extract DNA and sequence this DNA to reveal who is there and how do they survive to these extreme conditions. And we use a technique that the last lecture showed to you, that is the shotgun metagenome sequencing, in which we can describe these communities, not only who is there, but the functioning they are performing in these ecosystems. So we know if, for this scheme here, for example, if Alejandra is a pilot, so we can describe the function of the microorganisms living there, and through this metagenomic technique. And for this, we take this deep sea or volcanic samples, extreme marine samples. We extract the total DNA present in the samples, and we fragment this DNA in small pieces, small sequences of DNA, and we sequence these small sequences. And based on this, we can do bioinformatics for describing what are they doing in these ecosystems, their metabolic potential, and how do they survive under these conditions. For example, their molecular strategies to survive to high temperatures, low temperatures, or the parameter we are studying. And a very new technique we are using based on this metagenomics is to reconstruct entire genomes of these extremophiles without need to cultivate them in the laboratory. So with these small pieces of DNA, we use bioinformatic tools to reconstruct these genomes, and we can have hundreds of genomes, almost complete or draft genomes, to describe extremophiles that are not yet cultivated, and even describe new extremophiles, new lifestyles, novel species. So it's a very interesting tool that is very recent because of the developing of these bioinformatic tools. So the cave I mentioned, it is not a marine ecosystem, but we start with these environments, collecting in a region in Brazil, in the interior of Brazil, and where we have a lot of different quartzite caves, and we study this specifically and have some images of the sampling here. And we did the metagenomics and the reconstruction of the genomes based on this environmental DNA, and we could reconstruct several genomes of different organisms, different bacteria and archaea. And we also describe their metabolisms, and we observe that they are very versatile, probably because of the adaptive demand under this cave oligotrophic conditions. So we could observe, for example, oxidation of iron, methane, sulfur, ammonia, different energy forms, and reduction of several nitrogen compounds. And we selected 3 extremophiles of this cave based on these genomes. This one is capable of performing anaerobic methane oxidation. This here is able to resist to ionizing radiation, and the third is able to oxidate iron, potentially because they are this bacteria is still very poor describably. And these are probably new species or genera, and we proposed these 3 as novel functional models, specifically for the search for life on Mars. And for example, methane we know there is was detected on Mars and could serve as energy source for potentially life that could use this methane to obtain energy. That is on Earth, we call it as methanotrophs. So we also related to this genome information with cave mineralogy. So it was a multidisciplinary study, it was very interesting, and we published here in Astrobiology Journal. And just to have an example how we start to use these metagenomic tools for astrobiology purposes. So going to the marine ecosystems itself, here the first one we are studying is some active volcano in Antarctica. We have 4 active volcanoes in Antarctica, and this we are studying is Deception Island. It's an island that is a volcano, and it's a very interesting environment because we can find in the same environments all the types of extremophiles related to extreme temperatures, from psychrophiles adapted to low temperatures to hyperthermophiles adapted to high temperatures. So Deception is a very interesting environment when we can observe. Here is a view of the island, and it's actually an active volcano, the island itself. And we observe here a caldera. There is the volcanic caldera that is flooded by the sea. And in some regions, we can observe active fumaroles where we collect these samples. And we go to Antarctica, just to briefly mention that we go to Antarctica with the Brazilian Antarctic program to collect these samples with all the logistic of the Brazilian program. And we go to inside to this volcanic caldera with the vessel, with the Brazilian vessel, and to collect samples near these fumaroles that can reach 100 degrees. So it's very hot, and we collect these samples in a temperature gradient from hot to low temperatures. There is a very close, few meters of difference here of distance. And then we collect these sediment samples associated to fumaroles and sediment samples associated to glaciers to understand how life can support these extreme temperature gradients. And basically what we found is that the community is very different according to temperatures, as we expected. And what was very interesting is that the hottest fumarole was exclusively dominated by hyperthermophilic archaea that is common from the deep sea, as this pointed here. And we review some molecular mechanisms for survival under hot or freezing conditions. For example, fumaroles, we found a lot of genes related to oxidative stress or proteins that are adapted to these hot temperatures. And glaciers, we found specifically genes adaptation to, for example, osmotic stress or proteins that are specifically can respond to these low temperature conditions. And another thing that was very interesting is that these metabolic potentials, we found that the metabolisms were very specifically according to temperatures. Just briefly to explain, for example, in hottest temperatures, we saw a domination of chemolithotrophy, and on the other hand, in glaciers, specifically nitrogen metabolism. So we can understand here that the metabolisms were dependent on the environmental temperatures. And also we did some correlations of these microorganisms between each other and with environmental parameters. And one thing that was very nice is that we not expected is that the hottest temperatures saw more interaction between these microorganisms than the lowest temperatures. And we described different genomes of these extremophiles of this volcano and the energetic metabolisms, stress responses. We describe novel lifestyles and potentially novel species of extremophiles in this volcano. And it's very nice. This, if anyone is interested, can access this article that was published here. And another question we had was that if these extremophiles could be preserved during long-scale dispersion over Antarctic atmosphere. And for this, we collected samples from the interior of the Antarctic continent. Brazil has this model here that is called Cryosfera 1. And in this model, we could we did this like a pit structure to collect different layers of this snow. And we found a lot of marine groups in this interior of the Antarctic continent. So it could show to us that these marine extremophiles, or at least their genetic material, were preserved through these long dispersion patterns from the coastal regions of Antarctica to the interior of the continent. So we could observe that these extremophiles had their genetic content preserved through this travel through the atmosphere, the Antarctic atmosphere. And another environment we are studying with the astrobiological purposes is the deep Southwest Atlantic Ocean. So when we think about deep sea, a lot of people might imagine that it's a homogeneous place, but it's actually a heterogeneous place where we can find diversity of geological features in these different deep sea regions. And the geological feature we are studying is a carbonate ridge that was recently described, that is called Alpha Crucis Carbonate Ridge. Is the biggest ridge, carbonate ridge in the Southwest Atlantic, and it was described by geophysics, and we did the microbial perspective of this giant carbonate ridge, where we found different communities, different microorganisms from the top of the mount and the base of the mount. We found different groups in these different regions and in different depths. We collected the sediments in different depths, and we found, for example, in the deepest sediment layers, some archaea groups that are related to the origin of eukaryotes. There is called Asgard supergroup of this archaea. So it's a very interesting group we are studying now. And just an example that we can see that in this place, we observe almost uncultured and unknown extremophiles, especially in the subsurface samples. And we study also carbonate-rich sediments, surface and subsurface sediments in the region, also in the Southwest Atlantic. And we describe it, we can observe that the archaeo communities is totally different from the surface, from the deep layers. And the deep layers are dominated by diverse groups of archaea. Another geological feature we are studying in the deep sea are the pockmarks. there are deep depressions in the sediments that are created by the escaping of gas, especially methane, from beneath the seafloor. So we can see these big holes that are promoted by this gas escape. And it's a very interesting environment when we think about the Enceladus, that a lot of studies have describing the presence of methane clatrates in the plume of Enceladus. And this methane, as we found here on Earth, might be served as an energy source for the methanotrophs, so bacteria and archaea that can use this methane for obtaining energy. And we describe these pockmarks, the microbiology of these pockmarks by genomes from metagenomic data. So without cultivation, we describe several genomes, almost 65 genomes from these pockmarks. And again, we describe their metabolisms, how do they obtain energy from these ecosystems. We found a lot of diverse metabolic energy pathways from nitrogen metabolisms, sulfur metabolisms, or even methane metabolisms. We found a diverse metabolic potential of these microorganisms in these pockmarks. And our next steps now is based on all these extremophiles diversity and metabolic models in extreme marine ecosystems. The next step is to understand if can we propose novel biosignatures for the search for life in these ocean worlds based on this metagenomic data of uncultured microbes? And how can we do this? Because when we study these marine ecosystems here on Earth, almost all of these microbiome are not yet cultivated. So we need to use these tools, these metagenomic tools, DNA tools, to reveal their diversity and adaptation to this environment. So the cultivation is still very hard, especially for marine microbes, marine extremophiles. So our next step is to understand how can we propose these novel biosignatures for the search for life in ocean worlds here in our solar system. And just to finish, I think one thing is important to show to you that we are performing here in our university different science outreach activities, including these marine extremophiles. For example, we did this exhibition that we called Microheroes of the Oceans, to show to children in general, for example, bacteria and archaea as heroes and not as pathogenic or causing bad things to us. So to show them that these extremophiles and these microheroes, bacteria and archaea, are very important for our planet and then for astrobiology. And we are performing different activities inside the university or even in the streets of Brazil. And also we go to the schools, especially with this initiative that is organized by this association of polar early career scientists. There is APEX, and we promote the International Polar Week, in which the researchers go to the schools to talk about the researchers, their studies to the children with different ages. So it's a very exciting event to talk about our studies with these children in these activities. So I would like to thank again the opportunity to be here. And these studies I did when I was a postdoc in the Oceanographic Institute of the University of São Paulo in the Pelizari Lab. So if anyone is interested, can follow us here on Twitter. But recently I was hired as a permanent position as a professor here. And I'm coming soon. I will open my new lab of marine and polar thermophiles. So if anyone has interest in, please feel free to contact me. You can talk about partner possibilities. So thank you very much.